It's all yours.
There we go.
Yeah, it's yawn.
I'm here, I'm back. You'd think after all this Zoom, I'd have figured it out by now, but that's okay. Thanks everyone for joining today. I'm happy to be here, even if it's over Zoom. I know that's probably getting old at this point for everyone, but I'm glad you all made it. Hopefully I can make this entertaining. And I'm happy to take questions. I'll be taking them at the end, but if you think of something in the middle of my presentation, go ahead and type it into the chat, because I always end up forgetting my questions by the time we get to the end, when someone else is speaking, of course. So I know that that's helpful. But yeah, so I'll be talking to you guys about my research that I've been doing for the last 6 years here in Jupiter, Florida. But first I thought I'd start off by telling you how I got interested pursuing a career in science and kind of how it's transitioned into the work I'm doing today, and kind of the basic steps of what getting a PhD looks like. So if any of you are interested in science or don't really know what a PhD is, hopefully I can enlighten you a little bit that way. There we go. Okay, and now we figured out how Zoom works still. All right, so as Dr. Hazen said, I was raised in the Bay Area in California, about an hour east of San Francisco. And what's obviously nice about that, in addition to the nice weather, not unlike the nice weather we have here in Florida, is that I grew up near all this tech hub. And so where I grew up is kind of this white space out here where my red laser pointer is shown. But obviously, excuse me, very close nearby, there's all this tech up and coming. Obviously, you got Google, Facebook, Apple, got everything pretty much right there in the Bay Area. So the emphasis on learning about technology, science, mathematics, all of that was very strong. And so all of our schools were usually pretty good about emphasizing learning about science and math and a little bit of engineering here and there. But the city that I grew up in, called Livermore in California, is what they call wine country. And so it's a little bit south of Napa for any of maybe you older people who know what Napa Valley is and where all those wines come from. We're a little bit, maybe 2 hours south of that, but we grow a lot of grapes for wine. You can see these lovely vineyards. And each year we have a rodeo that comes by. And so all of our 4-H programs are really heavily invested in. So a lot of agriculture research is popular in this area, along with maybe your more standard technology and computer science that you might think of with California. We also have the Lawrence Livermore National Laboratory, which was about a 15-minute drive from my parents' home when I was growing up. And this laboratory focuses mostly on like defense research. So you think of biosecurity, intelligence, nuclear deterrence, as well as energy research. So you've got a lot of kind of chemistry, physics, and then the biology and agriculture. So you'd think being surrounded by all that science, I would have maybe picked up on it a little sooner, but growing up, I really wasn't that interested in science. I didn't find it that interesting, in part because, middle or like elementary through a lot of high school was just memorize this fact, regurgitate it on a test. I didn't find it that interesting. Instead, I was more focused on sports. So I did gymnastics for many, many years. And then in high school and in college, I transitioned into springboard diving. So I was really focused on that for a good portion of high school. But at some point, one of my friends decided to take a biotechnology class at our high school. And I had an open period, so she dragged me along to it. It wasn't something that I necessarily wanted to do, but I was open to try it since I had a friend to go with me. And basically biotechnology is the study of the technology that's being used and employed within the biomedical research fields. And so basically trying to study how to, fix human diseases and promote human health. And obviously we weren't doing anything too extravagant in high school. The school had gotten a grant to have this class wherein they were able to obtain some secondhand instruments such as these pipetters and some lab notebooks from the nearby startup technology companies near to us. And we got hands-on experience working with science tools and learning how to design our own experiments, all very basic stuff. We weren't, curing cancer or anything in high school, but it was just really eye-opening to me that all that information I had kind of memorized up until that point was actually very useful in a real world application. The teacher I had in high school had worked in industry for a while as a scientist, so she had a lot of really practical applications for why we would be memorizing this information, why would you learn how to pipette, that sort of thing. And is that hands-on experience that I really, really liked. And you'll see that theme throughout. I'm very much a hands-on sort of learner. If I'm not doing it, it's hard for me to learn. So I graduate high school and I decide I want to continue in science. This is now my newfound thing. I'm going to go to American University, where I did both springboard diving and got my bachelor's of science in biology. So I went all the way across to the East Coast. And at American University, it's obviously in Washington, D.C. In the springtime, it's really gorgeous. You have these cherry blossom trees downtown, by the National Mall. The campus itself is really gorgeous. And for those of you who are in high school looking at colleges, for me personally, this college had the best of both worlds because it was a medium-sized college, about 5,000 students, but the biology program was very good and mostly focused on pre-med, but it was very small. So we only had about 20 students in my graduating class. And so all of my, say, upper division classes where I was, learning about science and how science is done in the real world, we're only with a handful of other students. And so I really got that one-on-one experience with my professors, much more so than had I been in giant lecture halls. So for me, this worked. Obviously, everyone learns differently. But during my tenure at American University, I took a cell biology class from Dr. DeSico Skinner, who's this woman pictured right here. And I just really loved her teaching style, and I did really well in her class. And I went up to her and I was like, I love your research. She does cancer biology research. And I was like, can I please join your lab? And thankfully she had an opening at that point, and she was able to let me kind of try out for a few weeks, see if I liked it. And I ended up staying for about 2 years doing research in her lab, helping out where I could. So I mentioned that she does cancer biology research. So I'm sure most of you are aware, but to understand cancer, you have to understand how cells divide in order to understand what I was doing there. So a cell is the basic unit of life, and cells make up every living thing. You've got plant cells, cat cells, dog cells, tree cells, human cells. So when our human cells divide, let's say, just on a normal day, they divide from time to time as you grow taller, as you get older. Eventually, they will get damaged. Say, you're out in the sun and you get a sunburn, or maybe you cut yourself playing a sport, you have a damaged cell. And so if it can't be fixed, what will happen is that cell will have to go through a process called apoptosis, which is programmed cell death. And what that means is just that one individual cell dies and the rest of the organism, say the rest of the human, gets to survive and live another day. However, in cancer, what ends up happening is that damaged cell, the process that allows it to die isn't working correctly for one reason or another. And the cell ends up dividing over and over and over again. And if it divides enough times, it creates a tumor. And so tumors end up sequestering all the nutrients from the rest of the body, putting pressure on our organs, and eventually leading to the death of the person who has cancer. And so cancer is really, really difficult disease or set of diseases to solve, in part because what causes the damage and what causes the overgrowth can be different for each individual person. And so one kind of therapy that works for one person might not work for another. And so it makes cancer an extremely difficult set of diseases to study, to understand, and to cure. And so what we were interested in Dr. DeSico's lab was understanding the relationship between, say, fat cells in a human body and cancer cells. So no matter how your body is proportioned, everyone has fat cells. However, some people obviously have more. And unfortunately, what they found is that people who are obese are not only more likely to get cancer, but once they get cancer, they have a much poorer prognosis than someone who is not obese. And so we were interested in understanding what is it about the fat cells, and especially fat cells in someone who's overweight, and how that interacts with the cancer cells. And so we found a number of things, but that's not the focus of this talk. But what is fascinating is just this interplay between different cells and how they can talk to each other and lead to disease or to healthy states. And so what this research really showed and got me interested in is this: how do you go from having a healthy state to a disease state? And then hopefully one day scientists will be able to push that disease state back to a healthy state. So in order to kind of pursue this interest of understanding health to disease, I wanted to keep doing research. And so professors at American University suggested that I go off and get my PhD, which basically means you get to keep going to school forever. Hopefully not forever, but basically a PhD generally lasts 4 to 7 years. And unlike high school or undergrad, where there's a set number of classes you need to take in order to graduate, it's a little bit more open-ended. So you do enough research and you make enough of an impact, then you can graduate. So I'm kind of in that process of just trying to finish up all of my experiments. But 6 years ago, I came to Scripps Research in Jupiter. And so obviously came back to the warm weather I was more accustomed to in California than I was in DC. And I joined the lab of Dr. Katrin Karbstein, who is pictured right here. And these are my wonderful lab members who I get to interact with every day. And I'll get into more about her research, but it is looking at kind of the transition from health to disease and how to hopefully prevent it or cure diseases once they arise. But in order to understand my research, you have to understand a few basic things that probably will be a review for most of you, but just in case you fell asleep during this lecture of biology class, we'll review it, which is that obviously, like I mentioned, all cells or all organisms are made of cells. So you've got bacteria cells, plant cells, human cells look like this, or all eukaryotic cells generally look like this. And then within even one organism, there can be billions of cells. So if you look at an individual human, we're made up of billions and billions of cells. And these cells can be many different kinds. They can be heart muscle cells, or just, your normal muscle cells, blood cells, brain cells, you name it. Every organ has a different set of cells because they have a different job to do in your body. And the way that all of these cells can communicate with each other, form their structure and perform their job, such as, sending signals from different neurons so you can think and act, muscles in the heart and in your arms, so your heart beats, or your muscle, you can move your arms around. This is all due to protein. And so protein is this functional component within your cell. And most of us have heard of protein probably from your diet, right? Everyone says you have to eat enough protein, or maybe you're trying to go to the gym and get really strong like Popeye. You want to eat a lot of protein. However, the protein that we consume, whether it's from, land animals, fish, if you're getting it from, say nuts or beans, dairy product, what have you, this is all protein from other organisms. It's foreign protein, if you will, and it's not human protein. So we eat this protein, we digest it, and while we're digesting it, we break it down into its individual components. And so these individual components, you can think of them as little Lego pieces. You can take them apart, and then they're sent off to different parts of your body, to every cell in your body. And then each one of those cells can use those protein components, those little Legos, to build human protein within the cell. And they can build specific proteins that are necessary for that cell to do its job. So for, say, your muscle cells, you might need a different set of proteins than maybe your brain cells need. And so each cell can then use those components, build the proteins it needs, and be functional for whatever purpose it needs to be functional. And in a healthy state, every individual cell is able to regulate the number and different kinds of proteins within that cell. So let's say you take a heart cell and it makes very few of protein A, but it makes a lot of protein B, and that's normal, right? And so what it'll normally do is try and balance out, not necessarily so that every protein is made the same amount. Some proteins are higher in abundance, some are not as abundant, but that it's always made in the same ratios. And this helps to protect and support a healthy cell. However, if this protein balance is disrupted, so now you're making less of the protein that you need a lot of to keep your muscles healthy, and you're making a lot of the other protein that you're not supposed to make a whole lot of, and you're disrupting this protein balance, you end up with a variety of different kinds of diseases, including a number of cancers where this happens, a number of disorders related to aging, and many neurodegenerative diseases are caused by this disrupted protein balance, or homeostasis, as we call it. So in order to understand how this protein balance is messed up in your cells, you have to understand how are proteins made and how are their levels being balanced in your cells to maintain healthy versus diseased state. So proteins are made in our cell through the central dogma of biology, if any of you have heard of that. But basically, you start off with DNA, which I'm sure you've all heard of through like different DNA sequencing technologies with like ancestry.com to figure out maybe where you're from, or like who your ancestors are, where they came from around the world. But DNA serves basically as a blueprint of information. It tells your cells how to be a brain cell or a skin cell, but it doesn't actually do anything. It's not functional in that way. And so what DNA needs to do is it undergoes a process called transcription, and basically DNA is then made, or mRNA is made from the DNA through this process of transcription. And mRNA, or messenger RNA, is the thing that is going to be read to make protein. So messenger RNA is probably something you might have heard from all the COVID-19 vaccines. It's a component of 2 of the major COVID-19 vaccines. But mRNA is important because it is used as the template that is read into protein. And it's read by this thing called the ribosome. So the ribosome are these 2 big blue blobs in my cartoon, and it moves along the mRNA and basically reads it and turns that into protein, a big string of protein. So each one of these balls would be those little protein components that you ate, digested, broke down, sent to your extremities, and now your cell and your ribosome in your cell is building back human protein based on your human DNA, and therefore the mRNA. Now these individual ribosomes, so these blue circles here, are actually really complex. I call them protein making machines because, like any machine, it's made up of a bunch of little parts, and without any of those parts, it doesn't quite function right. So the ribosome, which we call an ADS ribosome, is made up of 2 different subunits, hence those 2 blue blobs. And within those blue blobs, we have 79 ribosomal proteins. So ribosomes make protein, but they're also made of protein. It's a chicken and the egg problem. So you have 79 ribosomal proteins. You have 4 ribosomal RNAs. So that same chemical that is read by the ribosome to make protein, something similar is in the ribosome and keeps its structure. And in addition, you have 200 plus protein assembly factors that help to assemble the ribosome, but they aren't part of the finalized ribosome. But they're very, very important to make sure that ribosome gets put together just properly to be able to make proteins. Now, when it's all complete, the ribosome looks like this if you were to look at it under a very powerful microscope. So the large subunit is called 60S, and it's here in purple, and the small subunit is called 40S, which is shown here in green. Now, you'd think with so many components, if I gave you, say, 83 different Lego pieces and said, build something, each one of you would build something different, most likely. However, the ribosome is made pretty much the same way each and every time. Despite having all those components, each and every time it's made the same way. And those 200 assembly factors do the same job each and every time. Now, these ribosomes, your human cells are making hundreds of thousands of them every minute. They're making a ton of them, and they're made the same way each and every time to keep you healthy, which is amazing. And like I said, the healthy cell, all the ribosomes are going to look the same. So now they're in these little gray blobs inside of your cell. And this helps to maintain that protein balance. Because the ribosome is reading that mRNA, it's making the protein. If the ribosome gets messed up, it won't be able to perform its job correctly. And unfortunately, we see this in a number of diseases. So many cancers have an imbalance in the number of ribosomal proteins that are being made. And so when the ribosome is being put together, it might miss a protein because there's not enough of it, or there might be a change in one of the proteins that makes it act differently within the ribosome. In addition, cancer cells oftentimes have maybe an overabundance of ribosomes. As you can see, there's more ribosomes in the cell than maybe in your healthy cell. And this helps the cancer cell, which is bad for us. It helps the cancer cell grow and divide, because as a cancer cell needs to divide, it needs protein. So it needs a lot of ribosomes to be able to keep dividing. And so because of all these alterations to the ribosome, as well as the very careful balance of ribosome numbers, you alter the protein balance in the cell, creating a sick cell. In addition, there are a variety of diseases that are all grouped under a class called ribosomopathies, in which the ribosome is, in which a patient is completely lacking a ribosomal protein or has mutations in either the ribosomal proteins or in one of those assembly factors that changes its function so it can't perform correctly. Unfortunately, ribosomopathy patients, who are often born with a variety of cognitive and developmental delays based on what part of their ribosome is broken, many of these patients are unfortunately at a much higher risk of developing cancer later in their life. So again, bad ribosomes, this disrupted protein balance leads to diseased states. So this leaves us with a number of questions. And our questions include, how do healthy cells assemble ribosomes? Like, how does this process even work under normal healthy cells? How does it work the same way each and every time? How do healthy cells, because inevitably things always go wrong. So if something goes wrong in a healthy cell, how does the healthy cell recognize that something goes wrong and then get rid of those bad ribosomes before it causes disease? And finally, when ribosomes are not made correctly, when they are misassembled, why do they cause the diseases that they cause? And how can we ultimately, change it to go back? Can we kind of reverse course, fix the ribosomes, fix the problem to create a healthy cell? Now, so I mentioned I was going to tell you a little bit about the process of getting a PhD. So I'm here, I've learned all about how cool the ribosomes are, how they're super important for preventing disease. But I want a PhD, so I have to do a bunch of research. So step one, you got to find a lab and a research project you want to do. So I found a lab, check, joined Katrine Cardstrain's lab. And then I have to choose a project. Now, when you choose a project, it can't be something you're going to do in a week. This is like, you got to think really big questions. So it has to be a project that's going to last for a number of years. Generally about 5 years is what you're aiming for. And so in order to choose a project, you have to understand what is already known in the field and identify open questions that still need an answer, something you're interested in, something that you think will help humanity, hopefully, or at least just keep you entertained for 5 years, right? So the question that I chose to focus on, although my project does touch on each one of these questions, is this first one. How do healthy cells maintain proper ribosome assembly? It turns out that up until about 10 years ago, we really didn't have much of a good answer for this. But over the last, well, I'd say 15 years, over the last 15 years, the field has really been growing quite a lot. So my research is adding to this. So to get to a hypothesis, which I'll get to, you have to understand a few more things about how ribosomes are constructed. So ribosomes are constructed kind of like on an assembly belt or an assembly line, where if you're going to make the same ribosome each time, you've got to have kind of an assembly order. This protein goes first, that RNA goes second, that protein goes third, that sort of thing. And during this process of assembling a ribosome, there are quality control checkpoints that occur throughout the assembly process to make sure two things. The first of which is that the ribosome is being built correctly. Structurally, it's intact. And two, that it will function correctly, that it will make proteins and it will not mess up the proteins. Because if you have a bad ribosome, we know that it starts doing weird things on mRNA, doesn't read the protein right, doesn't make the right protein. Then it disrupts that balance and disease happens. So you have to make sure that it's going to function correctly and that it's made correct and it's physically made correctly. If both of those things are good, it gets the green stamp of approval and gets to translate. Nice, pretty ribosome. However, if for some reason the ribosome fails a quality control step, it's not structurally intact, it's missing a protein, or for whatever reason, it's just not working, right? It's going to fail the quality control step and be sent for degradation, basically thrown in the cellular trash can, if you would. However, in a disease state, if you get misassembled ribosomes, what ends up happening is if, they might be correctly assembled sometimes, the ribosomes, and then they're allowed to translate, they pass just fine. But if misassembled ribosomes come up in a sick cell, they might not pass the quality control step, but they might still be allowed to translate because it either what's wrong in the disease state is that the quality control step is messed up, or maybe it fools the quality control step into thinking that the ribosome is okay when it's not. And so you end up with these misassembled ribosomes that still participate in translation, leading to that altered protein balance and the disease. So you can think of misassembled ribosomes in the same way that if someone's designing a car and then they forgot to put in the brake and you go for the test drive, you're not going to notice anything's wrong with the car until you want to stop the car. The car works just fine most of the time. It's only when you want to stop that it becomes a problem. So you can think of the same way. Misassembled ribosomes can still translate, but they're going to have problems. So getting into the nitty-gritty of ribosome assembly gets pretty complicated really fast, as you can imagine, with all those moving parts I have to get in. But ribosome assembly starts off in the nucleus, which is where your DNA is, where the DNA is going to be transcribed into RNA, which is this black squiggly line here. Then all of these proteins and assembly factors get loaded onto the ribosome one by one by one. And over the course of time and throughout a number of quality control steps, the ribosome is then exported into the cytoplasm for a very late stage of assembly. And eventually, once the whole ribosome is put together, all the assembly factors are gone. You have a mature ribosome that can bind mRNA and make protein. In my project, I decided one of the most important quality control steps that was not well understood was the very, very last one. How does a cell, or how does a ribosome go from being an immature, kind of pre-40S, if you will, ribosome into that mature ribosome that can translate? There's evidence in the field that ribosomes that look like this, that are not fully mature, mess up during translation if they get out there. So it's very important to be able to separate these ribosomes cannot bind mRNA, and these mature ribosomes can bind mRNA. So I wanted to know, what is the quality control step that checks the ribosome one last final time and then gives it the final green stamp of approval to become mature? All right, so what these ribosomes look like at this very, very last stage of ribosome assembly is you have this pre-40S, or this immature ribosome, that's in this gray blob right here. And it's bound to two of my favorite assembly factors, maybe not the ribosomes. The assembly factors are called PNO1 and NOB1. These two assembly factors are not part of the final ribosome. They have to be removed from the ribosome before it can bind mRNA and translate. In addition, these immature ribosomes have this extra piece of ribosomal RNA that has to be cut off, removed before it can translate. So at this point, this ribosome is not allowed to translate. It can't bind to mRNA. It cannot translate in a healthy cell. This won't happen. That's what the red light is for. Then there's some sort of process that wasn't well understood, whereby this protein NOB1 is basically a scissor protein. So it goes and it cuts that extra RNA. It's called an endonuclease. So it cuts the RNA, and then it's removed. That extra piece of RNA in red is removed, and then its little binding partner PNO1 is also removed from the ribosome. At this point, it's given the yellow light. And there's some sort of quality check that this all happens, and then it's fully mature and it can go translate. And so my big question was, what is regulating this quality control step? There must be something that's looking after this and be like, this has happened, it's now mature, it can go. And there's evidence in the field that it was due to a protein called RIO1. This is another assembly factor, one that binds for a very short amount of time and then fell off. They knew it bound, they knew it did something, but they didn't know what it did, and they didn't know really why it was important. So I thought this might be it. This might be regulating the quality control step. So I now understand what's in the field, and I know an open question that I could potentially answer. All right, so I'm going to form a hypothesis, design an experiment to address the hypothesis, adjust the hypothesis depending on how the experiment goes, and do it all over again. And you do that for 5 years, and then you can move on. So I will get to the short of it. I won't drag you through 5 years of work, but I'll show you a little picture of what 5 years worth of research might get you, or at least a couple days worth of research. So my hypothesis was RIO1 regulates that final quality control step, and it does this by releasing NOB1 and PN1 from the ribosome only after that RNA has been cut. So in order to address that question, we have to be able to design an experiment. And unfortunately, and fortunately, don't get me wrong, we can't experiment on humans very easily or morally. So we have to find a model organism to work with, or you have to work on human cells alone. Now, human cells are very complicated to work with for a number of reasons. So I chose to use yeast as my model organism. In lab, this is often called Saccharomyces cerevisiae or baker's yeast. Many of you will know this from bread, that really nice smelling baked bread. Those of you who are old enough to drink alcoholic beverages, as well as nutritional supplements such as probiotics that you might take to help settle your stomach if you have an upset stomach or if you're taking antibiotics. And it's also a good source of protein and vitamins. Now, in the lab, yeast are really very useful. They're a fungus. They're single-celled eukaryotic organisms. So they're similar to us in that they're eukaryotes, but one organism of yeast is one cell, whereas we are billions. So it makes it a lot easier in that way. You only have to deal with one kind of cell. And they double every 90 minutes, which is really fast because humans double like every 25 years, 30 years, I don't know, something like that. It takes a while for us to produce children. It does not take a very long time for yeast to produce children. They're much smaller than human cells. They're not very expensive to work with, and they have pretty simple genetics that you can work with. Makes it very easy to manipulate them. And nobody cares if you kill yeast. That's the important part. Nobody cares what you do to them. So you can manipulate their genetics and you can learn all sorts of things. But at the end of the day, who cares about yeast, right? We're talking about human diseases here. Why would I study yeast? Now, this is the kicker. Ribosome assembly in yeast is actually very similar to ribosome assembly in humans. It's a very well-conserved process. There are ribosomes. There are ribosomes in every single living cell, and it turns out that within eukaryotes, the process of making ribosome happens to be really, really similar. And so we can test out all of our hypotheses in yeast before we move into a more human cell or organism that's maybe more similar to humans. And so yeast in the lab looks like this. Oftentimes they'll grow as individual colonies, these little white dots, or you can streak them out onto agar plates and they look like this. Under a really high, powerful microscope, you can see yeast look more like this. And then you can also manipulate them to express different fluorescent proteins so that you can see how cells are moving, as well as how individual components within the cells are moving, which can be a really useful tool. Now, the lab that I've been working at Scripps, or I guess Scripps labs in general, look a little bit like this. So you have this really long hallway, and off to the left hand and the right hand sides are these long bays where science gets done. So this is what my bay looks like. And so I work kind of at the end here, but everyone has a lab bench and then usually a desk that you can work on. Oftentimes our lab benches are filled with notebooks, which you can see here, as well as boxes full of all sorts of reagents. We have all kinds of different apparatuses that we use and bottles full of media and liquid that we need to grow our yeast and do all of our experiments. So because we grow yeast, we have to use a lot of what we call incubators. So things that are held warm enough for yeast to grow, but not too hot that they die. So they like to grow at 30 degrees Celsius, which I believe is like 78 Fahrenheit, something like that. Anyways, it's warm, but it's not too warm. And so we have these shaky incubators, and oh good, the video is working. So you can see they just shake like this. It helps keep our yeast fully, all their nutrients swirling around, so they get access to all of their nutrients. And this is just more equipment that we use. All right, so back to the hypothesis. My hypothesis was that we're going to break it into two parts because there's going to be two experiments I'm going to show you. The first experiment will address the first half of the hypothesis, which is that RIO1 regulates this final quality control checkpoint by removing NOB1 and PN1 from the ribosome. So basically we're asking, does RIO1 remove these two proteins? The way I'm going to do that is called an in vitro release assay. So in vitro means outside of the cell. And so basically I grow up a bunch of yeast cells, and then I'm going to take their ribosomes out of their cells. Now, the way that I do that is I first crush up the cells and break open the cells because they have a cell wall. So you got to break them open and take out all the insides. Now, the insides obviously have ribosomes. Now, I've genetically modified these yeast cells to be lacking. So delta, that means they're missing RIO1. They don't have any RIO1. So what this does is it accumulates this ribosome intermediate that has both NOB1 and PN1 bound to it. And I put a tag, or basically a handle, on PN1, and I can chemically grab onto that handle and pull out all the ribosomes and leave all the other cell debris behind. So now I have successfully purified my ribosomes. I then am going to perform an experiment wherein I will either add or not add RIO1, as well as an energy source RIO1 needs to function properly called ATP. And ATP plus RIO1 added to these ribosomes will then be incubated for an amount of time and then put onto what we call a sucrose cushion, which is just a very dense sugar or sucrose liquid inside of a test tube. And we put our ribosomes, RIO1 and ATP, on the very top of it, and then we spin it really, really fast in a process called centrifugation. And this helps to pellet all of our really heavy ribosomes, and anything that is not bound to the ribosomes, say like RIO1, will be free floating in these little black dots. And so the idea is that we can take this light gray liquidy material at the end, which should not have any ribosomes. Anything that has come off the ribosome, however, such as free or released NOB1 and PN1, will be in the what we call the supernatant, or that liquid part. However, if we take just that pellet or that solid material at the bottom, that should contain all of our ribosomes and anything still bound to the ribosomes, including any bound NOB1 and PN1. And so we can effectively ask, if we add RIO1, do you get more free PN1 and NOB1 than before? Did it release it from the ribosome? And so the way that we did that is we performed our experiment that I just showed you in a condition where we had, this is just a control condition, no RIO1 was added. And so we would expect that no PN1 and no RIO1, no PN1 and no NOB1 has been released. And so we did a Western blot, which is just a way to visualize protein. And you can see so B stands for bound, so that bound fraction, things that are still on the ribosome, and R for released, anything that's been released from the ribosome. So on the bound fraction, you can see PN1 here and NOB1 here. In the released, you see very, very little amounts. And so when I quantify that, and so I ask, what is the percent of NOB1 and PN1 in the released fraction out of the total amount that's there? We can see there's very little NOB1 and PN1, maybe 5% or so of each, that is being released. So not very much, but a little bit. And then I have a control protein to make sure that my assay is working. And that's RPS8. And RPS8 stands for ribosomal protein of the small subunit. So this protein is embedded within the ribosome, and it should never be released. It should only be bound. And you can see a band here suggesting that RPS8 is in the bound fraction and nothing in the released. So the experiment worked. Now, I did the same experiment and I added in RIO1, but I did not add in its energy source ATP. So it can't really function super well. But just adding in RIO1, you can see that this band in the released fraction got darker for PN1 and for NOB1. And when I quantify it, you can see there's a slight increase, mostly for PNO1 being released in the condition where I add RIO1. However, if I add in that protein RIO1 and its energy source, ATP, we can see there's almost an equal amount of PNO1 in that bound fraction, bound to the ribosomes, as well as in the released fraction, those that have been released from the ribosome. This is similar to NOB1, although maybe not quite 50-50, but definitely a lot more in the released fraction here when I've added RIO1 and ATP than either in the previous conditions. And so when I quantify, you can see there's even more NOB1 and PNO1 being released when I've added in RIO1 and ATP. What this tells us is that we get a green check mark. Our hypothesis seems to be correct, at least our data support this hypothesis, that RIO1 is removing NOB1 and PNO1. Cool, we've answered half of our hypothesis. The other half of the hypothesis is, does RIO1 regulate this process and remove these two factors only after the ribosomal RNA has been cleaved, has been cut? All right, so now the question mark is really at this scissor level. Does it only happen after NOB1 has cleaved? So in order to understand that, I have to tell you about how ribosomal RNA works. So there are 4 ribosomal RNAs in total, 3 of which I'm showing you here. 18S, which is in the small subunit, and these other 2, 5.8S and 25S, which I won't talk about much, but they're in the large subunit. But they are made from DNA, just like all RNAs, just like that mRNA I showed you earlier. However, it's made as one long transcript, but these 2 subunits need to be able to come apart. So you have to be able, the ribosome has to then have each of these intervening sequences cut off. So this black bar here, the red bar here, and the 2 black bars here and here have to be removed and cut. And what happens when you do this is you end up separating each of these RNAs from each other. We're just going to focus on 18S RNA because that's the only one I'm going to talk about today. So this 18S RNA, just like in the previous slide here, how NOB1 is bound to this red loopy thing, I have it in red here. This is that extra piece of RNA that needs to be cut off. And the very final stages, we know that NOB1 cuts it off, but we don't know exactly when it does this. Obviously, it has to do it while it's still bound to the ribosome, but we don't know if RIO1 is somehow monitoring this process. Is it waiting? And it will only remove NOB1 after NOB1 is cut. And so NOB1 converts this pre-18S RNA with the red extra piece to the mature 18S RNA. All right, so to address this question, we're going to use something called polysome profiling, which will allow us to visualize our RNA cleavage. And the way that we do this is somewhat similar to what we've done before, in that I take a test tube, I fill it with a sucrose cushion, but instead this one's actually a gradient, which means that the amount of sucrose changes at the top and the bottom, and it's a smooth transition from not very much sucrose to a lot of sucrose at the bottom. Basically, what this means is that when I put all of my ribosomes and all of the cell debris from when I crushed them up earlier, I put them in and I spin it really, really fast through centrifugation. Instead of pelleting all the ribosomes to the bottom, I'm able to separate them based on their density or based on their size effectively. And what this means is that free proteins will be on the very top of the gradient. The small subunit will be next, then the large subunit. And then any what we call a monosome, meaning one ribosome, one 80S with both the 60S and 40S bound. Now, monosomes can just be free floating, or they'll be bound to mRNA, which I have shown depicted in this little red squiggly line. Or you can separate them from what we call polysomes, which just means multiple ribosomes on one mRNA. And basically, we can visualize this in the cell, and it has done before, where you have this long string of DNA followed by the RNA strings and all these little balls of ribosomes translating them. And these are polysomes, so we call it beads on a string. It's kind of what it looks like. And we use this machine, which looks a little old, but it works super, super well to separate this and visualize it. When we visualize, what ends up happening is we have all of the heavier cell components end up, I basically flipped this, and the heavier cell components are going to be on the right-hand side, and the lighter stuff is going to be at the very top, which is going to be on the left-hand side of the image. And we get a nice polysome profile or a nice trace. And so the higher the peak here, that means that there's more protein available there. And so you see free protein, anything not bound to the ribosome, is this first peak. And then we have the free 40S, the large subunit, that monosome or one ribosome, and then all of these polysomes. So multiple ribosomes bound to one mRNA. Now the first peak here is 2 ribosomes on one mRNA. And for each peak afterwards, you add 1 more ribosome onto it. So 2 ribosomes, then 3, then 4, and so on. Okay, so the way that we're going to answer this question about RNA cleavage and whether Rio1 has anything to do with it is we first took healthy cells, those with your wild type healthy PNO1, so totally normal healthy cells. And we asked, what does the RNA look like in these cells? And so we have a way to separate out the RNA by their size, and we can visualize it in a process called a northern blot, which is kind of like when we were visualizing the proteins earlier, but here we're just looking at RNA only. And we can see that this mature 18S RNA that should only be in the 40S ribosome is not in the free fraction here. It's only in those that contain that small subunit. And you can tell that there's hardly any band here where it should be only the large subunit. And you see again that 18S RNA, very strong band, very dark band, here in each of these fractions that have the full ribosome that's translating. However, this immature pre-18S RNA that NOB1 has to cut before it can translate. That has to happen, otherwise bad things happen. It's not a healthy cell. So because this is a healthy cell, that won't happen. And what we see is that this pre-18S RNA accumulates only in that fraction which has the 40S, because it contains not only the mature 40S, but also the immature 40S. However, you don't see very much of that same pre-18S in the fractions that contain mRNA, those that are translating. And that makes sense. This is a healthy cell. It should not have these immature ribosomes translating. That wouldn't make any sense. So when I quantify what is the percent of this pre-18S RNA in the fractions that are translating, in the translating fractions that are making protein, out of the total number or total amount of pre-18S RNA in that whole cell, you can see it's really low. It's only maybe about 3%, which makes sense. Very little should be translating. Excuse me. And so what we wanted to know is, in an unhealthy cell, what happens, right? So when you mess with this particular quality control step, what happens? Now, a lot of people, when they get cancer, end up getting their genome sequenced so they can figure out what part of their DNA has been mutated or changed that has led to the cancers. And unfortunately, a lot of patients end up accumulating a number of mutations within this protein PNO1. Okay, and so that's where, we're looking at a structure of the ribosome here, and I've basically zoomed into this portion in this black box here, zooming in on NOB1 and PNO1. NOB1 here is in this blue cyan color, PNO1 is in kind of purpley pink, and then all of the cancer mutations are either in this green sphere or yellow. Now, one person did not have all of these mutations. This is a combination of a bunch of mutations from a number of different individuals. And so what we did is we designed a similar mutation in yeast based on some of the human cancer mutations we've seen or that have been noted in the literature. All right, because we're trying to understand, what is it that goes wrong in human cancers when you have this mutation, right? It's not just like what happens in yeast, but like what happens when human cancers get involved? What happens in their cells? What are their ribosomes doing? Why are they messed up? Why do they have cancer? So we take this cancer mutation in PNO1. We did a bunch of experiments, and what we found out is that it allows for the bypass of Rio1. Basically, Rio1 is no longer needed in these cancer cells. And so PNO1 with this mutation in yellow and NOB1 fall off the ribosome on their own. They don't need Rio1 anymore because of this mutation. Now, so the question becomes, what happens to that 18S RNA if you no longer need Rio1? Does that pre-18S RNA stay pre-18S RNA, or is it also cleaved? And this would tell us if Rio1 is somehow monitoring this process and would normally remove them only afterwards. I'll explain. So we did the same experiment. We took the cells that had that cancer mutation in PN1 and did the same polysome profiling. Now you can tell just based on the profiles themselves, they look different, right? You have very few polysomes. They're not translating super well. Clearly something is going wrong. So you look at the 18S RNA, it looks very similar, which we would expect that any mature 18S RNA is going to be translating. Fine. But in cells that have this cancer mutation, we see that this pre-18S RNA is not only in this 40th fraction, like it is in the healthy cell, but a lot of it ends up in this monosome layer in these two fractions here, as well as in the actively translating pool out here. We see a lot more bands. And when we quantify the healthy versus the cancer mutation amount of 20 or pre-18S RNA, we can see that a lot more pre-18S RNA accumulates in these in the translating pool of ribosomes. So it's not just sequestered into this state where it can't mature. Now they're binding mRNA, they're translating. They're not translating correctly because we know that these pre-18S RNA containing ribosomes move that balance of proteins. And so it supports a diseased state. And so our hypothesis, we're basically able to answer this. Now, the reason I'm able to conclude this is because when we bypass RIA1 with that cancer mutation, you now get, let's see. Okay, I'll get there. Okay, anyways, we're able to show this. So what I was able to show through these experiments is that one, RIA1 is able to license ribosomes to translate mRNA into protein, right? So normally RIA1 looks at the ribosome, says, yes, NOB1 has cleaved successfully. It's removed those two proteins, and now it gets the green light to go ahead and bind mRNA and make protein. However, under normal conditions, a healthy cell that is lacking RIA1 or something else goes wrong and RIA1 says, nope, this ribosome is bad, it will not allow the ribosome to translate. Very little of this ribosome will be able to bind mRNA and translate. However, if you have a cancer mutation in PN1, what ends up happening is you don't need RIA1 anymore. And you remove these two proteins, and sometimes that happens before NOB1 can cleave its RNA. And you end up with an immature ribosome that still has that red pre-18S rRNA bound. So what this means is that RIA1 is responsible for making sure that these immature ribosomes that have the RNA do not translate, because these ribosomes are going to disrupt the protein balance and cause a diseased state. All right, so I've formed my hypothesis, I've tested it, I now have a working theory of what I think is going on, not only in a healthy cell, but what's going wrong in certain cancer cells. So now I need to share my findings. So obviously I'm sharing it with you guys today, but this is an important part of any kind of scientific research, not just a PhD. Once you do all your experiments, you have to tell the world about it, whether that's going to scientific conferences or publishing in journal articles or, sharing your research with your friends and family. It's really important. And so that's basically what we've come to, is that RIA1 is really important for this quality control step. Now, this is a very tiny step in the grand scheme of things within ribosome assembly. So if you get anything from my talk today, what I want you to remember is that misassembled ribosomes that have been incorrectly built, can lead to a disease state where the protein balance is off, and now you can lead to a cancerous environment. And so it's really, really important for scientists to understand how ribosomes are normally constructed in a healthy state in order for us to maybe one day be able to cure or at least prevent some of these diseases. And hopefully transition a cancer cell back into a healthy cell, or target a cancer cell via therapy to try and kill it and leave you with only your healthy cells. All right, so finally, if you're interested at all in what I've done, I do have a paper out, you're welcome to read it, but you can also just look at either the Karpstein Lab website or Scripps if you just want to see what kind of research is going on in your neighborhood, especially for you high school students or undergrads. There are research opportunities here at Scripps, and any, like not just at Scripps, but a lot of research universities have that. And I've benefited so much in my journey to getting here and getting a PhD to hands-on research. So if your school doesn't provide any opportunities for hands-on, lab experiments, maybe in class or something, and you're interested in this, definitely check out the local universities or research institutes nearby, and hopefully you can find something where you can maybe intern or just learn more about the research going on. So just wanted to acknowledge, obviously, science is not done in a vacuum. So all of the support we get financially from the National Institutes of Health, as well as the Howard Hughes Medical Institute, my mentor, Katrin Krupstein, obviously Scripps at large, and Dr. Herzen for organizing this and inviting me here today, and all of the Meet the Science lecture supportees. And thank you guys for all listening. And I'm happy to take questions.
Fantastic, Melissa. I really enjoyed the talk. And again, a reminder to everyone, you're all muted, but if you have questions, you can type them into the chat window. And I'll give you a couple of minutes by just asking a question myself. So if immature ribosomes exist in these kind of diseased cells, and it's because Rio1 is not there, what is the thinking about introducing Rio1? Would that work to introduce it later to fix the problem? And or what's the thinking, to get rid of these immature ribosomes that sort of slip through the usual quality control?
Yeah. So obviously, I presented this in a very streamlined manner, and unfortunately, in cancer cells, these mutations are not the only mutation that these patients have. So they oftentimes have a mutation, say, in the ribosome, and actually some part of the ribosome is frequently mutated. But they also have problems in a lot of other things, including that degradation pathway. So at the beginning, I showed normal, healthy division. If a cell gets damaged, it'll get degraded or gotten rid of, it'll be killed. And cancer cells don't do that. That's because the pathway that would lead to it is generally one of the things that goes wrong in cancer. And so the thinking with ribosome assembly and trying to target it is that kind of like with a lot of cancer therapies, if you're ever targeting a cancer cell looks just like a normal healthy cell to any kind of drug or things like that. And so a lot of therapies, that is an option. People are starting to look into how do we modify the ribosome assembly pathway to, say, fix cancer. But a big problem with that is how do you target a specific cancerous ribosome or a specific cancer cell, and how do you distinguish that from anything else? So it is definitely an option to, say, add back in Rio1 or get rid of some of these more problematic proteins, or at least, say, decrease their level. So I remember I said the protein abundance gets messed up. You can lower one of them, raise the others. But I guess long story short, some people are working on it, but it remains kind of a far out hope at this point.
Yeah, no, sure. It's a multivariate challenge. I was just wondering whether technically it would be possible to introduce Rio1 into cells, and would it harm healthy cells if there was too much of it?
Yes. So from my experience, what happens if you put in too much Rio1 is you end up kind of getting Rio1 introduced to ribosomes too early, and it will actually kick off NOB1 and PN1. If there's too much of Rio1, it'll kick it off too early. So having too much of it is really bad, and not having enough is also bad. And so it really comes down, and frequently in cancers, sometimes you do get that disrupted balance. Oftentimes you're getting mutations that disrupt its function. And so rarely are you seeing it go away completely. Most of the time it's actually, say, overdone. There's too much protein, where you get mutations that act like it's not there because it's not functioning correctly. And so if you can put back in, say, a wild type Rio1 rather than the mutated version of the cancerous cell, that might be an option to help balance it out. But again, it becomes really precarious because you need to have it at just the right amount. So technically it can be challenging, but there are ways to get it done, and it'd be hard in a full organism.
Okay, very cool. So I don't see anything else in the chat. I guess our audience is still digesting the ribosome quality control systems, and hopefully they took some notes. And if they have any questions, they can obviously reach out through their teachers to me. I can pass it on to you if there's anything in particular they're interested in. So let me just thank you very much again for volunteering to share your scientific research with us and the whole audience. We did record it, so I send you a copy of it later so you can keep it in your archive. So when you get to my age, you can look back at it and see how you developed different presentations over the length of your whole career. So might find a little anecdotal mentioning later on. And so I think everybody enjoyed it. I just see at least one comment here. The others might be already distracted to do other things. But if anybody wants to express their thank you, please do it in the chat box. Melissa can actually see that too, I think, since I share the screen. So thank you very much again. I will follow up. I'll be in touch again fairly soon. I have a little thank you gifts that I need to get to you. So we'll email about that. So thanks again. Have a good night. And
thanks, everyone.
All the best with your research. And
thank you.
Let us know when you defend your PhD. And
definitely.
Then next time around, it's going to be Dr. Melissa Parker.
Yeah, and you've all had a primer now, so you can really jump into the research at the PhD level, I'm sure.
Okay, very good. Well, thank you very much, and bye-bye now.
Bye, everyone.